Hello everyone, welcome to our channel. Today we are going to discuss the genetic disorders. As we know, in our cell we have a nucleus. A nucleus is composed of chromatin composed of 23 pairs of chromosome in the somatic cells, 22 autosomal chromosome, 22 pair of autosomal chromosome and 1 pair of sex chromosome. In male, we write as 23 pair and sex chromosome X and Y, and in female 23 and sex chromosome XX. This is a diagrammatic representation of a chromosome in which the small arm is denoted as P and the long arm denoted as Q. Chromosome composed of DNA. The central structure is known as centromere, and the end of the chromosome have the telomere. They help in the cell division. How to write or read a locus, which is a physical position of a gene or a particular segment on a chromosome. For example, 15q13.3, in which 15 represent chromosome number, q represent the long arm, 1 represent the region, 3 represent the bend, and last 3 represent the subbend. So how we read? So this locus present at the long arm of chromosome 15, region 1, bend 3 and subbend 3. This is the location of this sequence. Now we will discuss about some basic definition that we should know before reading the genetic disorder. What is gene? Gene is a sequence of DNA which carries some information. For example, P53 gene and RB gene. What is a locus or loci? A specific physical location or position of a gene or a significance of chromosome, significant sequence of a chromosome. What is exon? They are the segment of a gene contain coding information. What is intron? Intron are non-coding gene segments. They are introns between the exons. Now we will see about the mutation. What is mutation actually mean? It is a permanent change or alteration in DNA sequence of a gene. And epigenetic, what is epigenetic? As the name suggests, above the genetics. These are the heritable changes in gene expression without changes in DNA sequence. They can be driven by the chemical modification or histone modification and result into the gene turning off or turn on. The chemical modifications seen in epigenetics are methylation, acetylation and phosphorylation. What is genomics? Genomics is a comprehensive study of a genome, and genome is a full complement of a genetic material in an organism, coding plus non-coding genetic material. Proteomics is a study of proteome which examine the protein structure, function, interaction and expression. Proteome is entire set of proteins that is or can be expressed via genome, cell, tissue or organism at a given time under a specific condition. So proteome is a dynamic, and unlike a genome which is static. So proteome is dynamic because it alters according to the cell type, developmental stage, and the environmental factors. And the study of proteome is known as proteomics. Now we will discuss about the basic classification of genetic disorders. So in genetic disorders we have three categories: single gene disorders, chromosomal disorders, and multifactorial disorder. In single gene disorder we have two subcategories: Mendelian inheritance disorders follow Mendelian inheritance, and the disorder with non-Mendelian inheritance. In Mendelian inheritance we have autosomal recessive, autosomal dominant disorder, X-linked recessive disorder, X-linked dominant disorders. In non-Mendelian inheritance we will discuss mitochondrial inheritance, genomic imprinting, triple nucleotide repeat disorders, and germline mosaicism. In chromosomal disorder we will discuss about the aneuploidy, maybe trisomy or monosomy, structural defects like deletion, translocation, inversion, or isochromosome formation. In multifactorial genetic disorder, as the name suggests, there is defect in gene as well as the effect of environmental factors. Examples of these kind of disorders are diabetes mellitus, coronary artery disease, hypertension, gout, cleft lip, cleft palate. So this is the basic classification of genetic disorder. Now we will see the mutation and some basic principle involved in mutation. Mutation is a permanent change in DNA. If the mutation occur in germ cell, so this will result in inherited diseases, and if mutation occur in somatic cell, this may result into a congenital malformation or cancer formation. Effects of gene mutation. So what are the general principle we know before discussing genetic disorder? Say in the category 1, point mutation within the coding sequence. Single base is substituted with a different base in point mutation. Point mutation can be missense mutation or nonsense mutation. In missense mutation, base replaces the normal base which encode different amino acid. If the substituted amino acid is biochemically similar to the original amino acid, then this is known as a conservative missense point mutation. If the substituted amino acid is biochemically different from the original amino acid, then this is known as non-conservative missense point mutation, which is seen in sickle cell anemia, in which thymine replaced by the adenine and glutamic acid replaced by valine. In the nonsense mutation, a point mutation in which result into the a chain terminator codon, which is seen in beta thalassemia, in which a cytosine replaced by the uracil and result into the UAG stop codon, and this will result into the chain termination. No protein formation is there after the stop codon. Mutation within the non-coding sequence like promoter and enhancer sequences, they are responsible for the DNA transcription. If the mutation occur in promoter and enhancer sequence, this will result into the decreased transcription. Point mutation in introns, as we have discussed, introns are the non-coding sequence of a gene. This will result into a defective splicing of the intervening sequences, and there will be no translation, so there will be no gene product synthesized. Deletion and insertion. As the name suggests, deletion of some portion of a chromosome is deleted, and in insertion some portion of a foreign or a different chromosome is inserted or added. If the number of base pair involved is 3 or multiple of 3, then the reading frame remain intact and there will be a gain or loss of amino acid. In deletion there will be a loss of amino acid, and in insertion there will be a gain of amino acid. But if the number of base pair involved is not 3 or in multiple of 3, for example, if the inserted base pair are 2, 4 or deleted, likewise the deleted base pair are 1, 2, 4, so this will result into the frame shift mutation, which is seen in cystic fibrosis, Duchenne muscular dystrophy and Tay-Sachs disease. Alteration in protein coding genes other than the mutation. As we know, mutation is a change in DNA sequence, but there are other alteration in protein coding genes like amplification, deletion and translocation. This may also result in a genetic defect. For example, translocation of chromosome 922 will result into the formation of Philadelphia chromosome, and this defect seen in the development of chronic myeloid leukemia and in cases of some acute leukemia. Alteration in non-coding RNAs, microRNA and long non-coding RNA. They are responsible for the regulation of gene expression. Trinucleotide repeat mutation, amplification of a sequence of 3 nucleotides. They involve guanine and cytosine in all the affected sequences. For example, Fragile X syndrome. Trinucleotide repeat mutations are dynamic and amplification increases during the gametogenesis. We will discuss all these disorders in detail later on. Thank you.